Hi, I'm Kala here.
In this video, we are going to explain about biological process of genetic transformation from Agrobacterium, Tumefaciens and viral vectors.
So, Agrobacterium tumefaciens is actually a natural tool for the plant transformation, where the DNA from the bacteria is transformed to the plant so that the DNA can be integrated into the genome of the plant. So, where the bacteria's DNA will bind with the hostone and will be integrated into the nucleus of the plants. So, it's also known as the agent of crown gall disease, where the Agrobacterium tumefaciens causes the crown gall disease by first transferring its DNA into the openings of the plants. So after it enters into the plants, it integrates itself into the plant's genome and causes the formation of this gall. So it works. The working of the Agrobacterium tumefaciens is actually by called as the tumor induction, where just the transformation of the DNA into the plant cell and integration of the DNA itself to become a part of the genome of the plant.
Okay, let's look at the biological process of genetic transformation from Agrobacterium to Tumefaciens in more detail.
The bacterium has a loop of DNA called the tumor-inducing or TI plasmid. A part of this TI plasmid will transfer into a plant cell. The DNA transfer induces plant cells to make food molecules for the bacterium.
Agrobacterium, which is present in the soil, start to communicate with plant cell and a wound on the plant body. The wounded plant release phenolic compounds that, depending on the species involved. In the cell envelope, a protein called VRA detects the chemical signal from the wounded plant. The phenol compound which was released by wounded plant will bind with VRA. The binding activates VRA, which can then activate another protein called VRG through a process called phosphorylation. Then, a number of genes called Vir genes are located on the tumor-inducing plasmid, where the genes encoding VRA and VRG are turned on all the time. The expression of the either Vir genes require activate VRG as a transcriptional activator. Two of the gene products are VIR-D1 and VIR-D2.
The VIR-D proteins are endonucleases, which is actually an enzyme that split at the 25 base pair sequence repeats at the end of the tDNA. As a result, cell produce a single-stranded tDNA molecule attached to VIR-D2 at one end. VIR-D2 directs the transfer of the tDNA into a plant cell in a process similar to bacterial conjugation. A number of genes are encoded in the tDNA and become expressed in the plant cell. Some of the gene products are responsible for the synthesis of the plant hormones such as cytokinin and auxin, which trigger the plant cells to divide rapidly and form an undifferentiated mass known as a crown gall tumor. The tDNA also encodes genes that control the production of unusual amino acid derivatives called opins. In return for delivering the tDNA to the plant, the bacterium gets the opins as a source of carbon and nitrogen. Researchers employ Agrobacterium to genetically engineer a wide variety of plants. In this procedure, the tDNA is placed on another plasmid that is more easily manipulated, and the DNA between the flanking sequence repeat regions is replaced with a gene of interest. Upon infection, the virulent system induces transfer of the tDNA to a plant cell without any tumor-inducing genes, allowing genomic integration of recombinant gene without tumor induction or opin production. For Agrobacterium Tumefaciens. Now let's discuss about the biological process of genetic transformation from viral vector.
Viral vectors are tools that commonly used to deliver genetic material into cells. It do not integrate into plant cells or host genome. Viruses have evolved specialized molecular mechanisms to efficiently transport their genomes inside the cells they infect.
Okay, now I'm going to explain about viral vector for gene transfer. So this is the natural virus and this is the gene of the virus. The gene will be removed for viral replication.
Once the viral replication done, therapeutic gene will be inserted. Now the virus is ready to infect a plant and the therapeutic gene will be delivered to a target cell. This is how the genetic transformation process occurs from viral vector and also in the Agrobacterium Tumefaciens.
Sekian sahaja daripada kami. Terima kasih.
Nandri.